the second day. Uh, Here I will share my screen. Today's topic is very much applied and very much specific. That what is the utilization of the entomopathogenic nematodes? Because yesterday we have seen that and many of the times we always see that the nematodes are always been considered as the enemy of the farmer, or it has always been attacking on the plants and causes the damage to our plants. There is economic damage. There is losses caused by these nematodes. But every time it is not true, because every cloud has its silver shining. Just what we say. Same is happens within case of the nematodes, where uh, some of the nematodes are also our friendly nematodes, and that we can use them against the for our control of the insect pest. So I will share the screen, sir. Slides दिसतायत का? Slides whether slides are visible?
Yes, slides are very much visible.
Okay. So yesterday we have seen the basically the nematodes are of two types. Basically, they are they parasitic to the plants. They causes the damage to the plant root system, and there is a tremendous loss caused by the nematodes. Our policymakers and those who are designing the programs at national and the state level, we should convince them that this is another constraint in our crop production. Alone nematode causes, because I think the estimate in 2016 they have one published one paper, and they have shown the estimate losses caused by the nematodes alone in crop production is reaches up to the more than 1,000 crore rupees annually in India. So, and again, it is different in different crops. Some of the crops are so valuable for us. Like example, in case of banana, example in case of some of the flower crops, some of the vegetable crops, which are very, very specific to us, where nematode damage is more, and they are especially they causes the tremendous losses in a particular cropping system. But today we will see that the entomopathogenic nematodes, particularly where we can have these nematodes, their role in the let's say for biological control, we can use them. These nematode, they're all behavior strategies, they're all adaptations what the nature have bestowed a gift, just like a gift to them to control the insect pest, and particularly all the major and minor pest of the crop plant. So today we will see what is their role, how they are actually we can utilize them, and who has introduced this, what is the pathogenicity, what is the mode of action. Everything we will see in this lecture. So can anyone please tell me how much percentage of losses caused by the insect pest in crop production system? Can anyone? Because you are all from crop protection background, so please anyone, please tell me that how much percentage of the losses caused by the insect pest alone in the cropping system? Can anyone? Hello?
So 15%.
15%. Can anyone have another figure? Yes,
Sir, 30 to 35%.
30 to 35%. Okay. Can anyone have another figure? Why someone is saying 15%? Why someone is saying 30%? Why can't we only stay on one figure? Or why can't we only have the figure which say only that this is the percentage of average losses caused by the insect pest? Can anyone have any different figure apart from this 15 and 30%? Please speak up, because if you are not speaking, then how you will able to understand? Yes, anyone? And how much percentage the percentage the losses caused by the diseases? Yes. You cannot rely on one figure, or you cannot have the figures which are only that, let's say the 15% and 30%. I will give you some of the example before introduction of this topic. Let's say in case of wide group, because where we are, I'm working actually, the wide group in sugarcane. In Maharashtra alone, we have seen that only one pest causes 80 to 90% losses caused by this pest in sugarcane. Whole crop plant was devastated by this pest. So this is one example I'm giving you. Let's take an example of fall armyworm in maize. Total crop losses 100%, same percent loss, same percent crop damaged by the insect pest. So you cannot just rely on one figure that's okay, this is the 15%, this is 30%. It is again vary according to the crop produce value. It's again vary with the cropping pattern. It again vary with which crop and which pest they are actually causing. And that is why because of these differences, because some of the pests are causing the 60%, like a sucking pest. Let's say take an example of sucking pest. In case of the aphids, thrips, jassids, white fly, woolly aphid, you can see if you measure the damage caused by this pest is an enormous in a quantity. You cannot even quantify the damage caused by this pest, because we don't know exactly what numbers they in what numbers they are present on our crop. So for control of this pest, and yesterday I have specifically, so I devoted some time for the population dynamics. So population dynamics and decision making in a pest management is the most important. So population dynamics means how much the numbers are there, how much numbers of insect, how much numbers of propagules, how much number of nematodes are there, and when we have to take the decision to control them. So in this situation, yesterday also I have said, and I'm again repeating, because of this wrong mathematics in everybody's mind, we are using the agrochemicals in a tremendous and enormous quantity. Particularly in sugarcane, or let's say take an example of grape. How much quantity of pesticides and how much quantity of insecticides which we are voraciously applying, and which we are actually polluting. It is not only to read only in the literature that this is causing the pollution, this is causing the contamination in our environment. If you go outside your home, if you go outside your campus, you will see that we are actually polluting our environment. How much quantity of one chemical, that coragen, that is chlorantraniliprole, how much voraciously people are using? Then what is the effect is that we have to change the strategy. Number one, insecticides, use of the insecticides is improper and indiscriminate manner. What happens? First is contaminating our environment, and secondly, the pest resurgence. Yesterday I also discussed about the pest resurgence. That means the status of some of the minor pests, the pest which we have never seen in our cropping system. Nowadays, those pests are coming up in a huge quantity. They are attacking in a huge quantity. I want to just share one of the story of just 2 days back. The wheat, we have here one of the trial, all India coordinated trial on wheat. We have here in our at place. Just 2 days back, the scientists came from the all over the India to visit our wheat plot, and we have seen that the pink borer, which is a stem borer, which is a normal pest which we particularly found in the wheat. Nowadays, there is a sucking pest is increasing in the wheat. Now, when we were doing the BSc Agri, when we were doing our graduate, we were used to taught by our teachers that wheat and rust. Only this mechanics was also only this combination was there. So wheat and only having the Pakshinia, PTC or Pakshinia, the fungi was there. Nowadays, this whole thing has been changed. So why it is changing? It's number one is the climate change. Second one, we are using the chemicals. We are using the not only insecticides, but nowadays the fertilizers, nowadays the spreaders, stickers, and adjuvants. Everything it is in such a combination matter. You cannot only now rely that only 15 to 30% losses caused by one of the insect. Any insect on any crop causes a lot of damage. And what is the strategy you use? Yesterday I told about the integrated aspect, and among that, the biological control is the most promising aspect. Now, what is important in biological control? The first one is that biological control is a very much safe to the other environment, which is 100% sure. Whatever biocontrol agents we are applying, whatever you know, you might be knowing about the trichogramma, trichoderma, pseudomonas, specilomyces. Let's take an example of metrorhizium, vivaria, whatever the fungus, whatever the bacteria, whatever the nematodes which we will see. First thing, I'm 100% sure they are safe to the other non-target organisms. Not just like the insecticides, they are very much highly toxic to the non-target organisms. Number one. Number two about the biological control is that it is a natural control. We are actually employing these agents to go off and to attack on their target pest. Second thing, which is a very, very important, because this is a natural fight, let's say. We are just igniting the fights between them. Let's take an example of trichogramma. I think everybody knows about this trichogramma. We are also in this lab, we are producing trichogramma. We are giving these cards to the farmers. What is a trichogramma? It is actually attacking on the eggs of the Lepidopteran species. Very easy, very, what should I say, very amenable biocontrol agents, very sturdy biocontrol agents to adapt them in many situations. Now you can see, instead of we are targeting to the larva, which is a damaging stage in most of the Lepidopteran space, we are targeting on eggs. So once the eggs is targeted, that means the further life cycle can be hampered, further life cycle can be stopped. So such kinds of biocontrol agents we have employed, we have tested, we have studied, we have in India, this research is continuously going on, and every biocontrol agents have their promising actions. So this is very much important. So first you must remember that first we should have confidence in our biocontrol agents, that whether my biocontrol agents have that much of capacity to control the insect pest or diseases or not. So biocontrol is one of the very much best strategy that we can have the promising actions. Number three, they are not causing any environmental pollutions. They are not causing any kinds of groundwater pollutions, whatever the pollutions and whatever the types of the pollutions is there. So they are not causing. And fourth important aspect about the biocontrol agents, and I think you have heard and you might have read in your books, that they are recyclable. We can use reuse also, because their populations once entered into the nature, once entered into the ecosystem, they are able to reproduce themselves, they are able to propagate, proliferate themselves on the field. So among these biocontrol agents, several aspects are there, like fungi are there, bacteria are there, some of the protozoans are also, the insects also we can use as a biocontrol agents. And among that, the nematode is another most important aspect. And why nematode is the most important aspect? Because nematodes have the very much specific targets to the larva of the soil-borne insects. Now, what are the soil-borne? they have their pupal stages in the soil. Many of these RB worms, Sporoptera, and let's say from these the families of the Noctuidae family, which is mostly the families from uh, nocturnal insects. They have their life cycles. Some of the stages of their life cycles are close to the soil. So where we have the one aspect to control the nematode, control the insect pest with the help of nematodes. Very promisingly, the EPNs, in short, it is known as EPNs, we have used in our insect pest management program. So today we will see, and today we will have the discussions on it. Now, are you able to see the slides? Hello, please respond.
Yes, sir. Yes, sir.
Now, we will see, and very briefly we will go. There is introduction and history, because who has introduced it and how the historical developments has taken place in this intro-pathogen nematodes. We will see the biology, we will see the pathogenicity, we will see the hits of host ranges, we will see the merits, demerits, and we will go for the conclusion, because it's a very short topic, but it's very much applied topic. I'm working on this topic because my research work is on this topic. I'm working again on the management of white grove with the help of the EPNs. So it's very topic very close to me. So don't mind if I speak somehow more on this topic, because it is very much topic which is very close to me, and I will try myself to introduce to help you to understand the topic, whatever the topic, whatever the information about the EPNs which I understood from my whole of my whatever the I studied as MS and PhD there. So it's a very important is here that this is a entomopathogenic nematodes. This is the nematodes and bacteria is the combination. So first understand, and I think I need a response from you all of you, that please can anyone tell me what is a mutualism? What is a symbiosis? Please, anyone, please respond. Whatever you have learned, that what is a mutualism, what is a symbiosis? Can anybody have any definition? What is symbiosis? What is mutualism? Yes?
Both the organism beneficial for each other.
Both the organism beneficial from each other. Anyone, any other, if you want to add what the Jeevan Savarkar has, what he has said, can anyone wants to add into this, what is the symbiosis and what is mutualism? Hello? Can anyone wanted to say something on it?
It is a close association between the organism that both the organism benefited each other.
Yes, of course, they are benefiting with each other, but can you give an example into this?
Like in mycorrhiza.
Yeah, what kind of association?
Like in mycorrhiza.
What kind of association they have?
Mycorrhiza having a fungi and a root of higher plants, and like it is a mycorrhizal association between fungi and algae.
What is the benefit for us? If they are making alliance, what is the benefit for us? Is it beneficial to us? Suppose A organism and B organism, let's say, both are wanted to join with each other. Let's say they wanted to join hands with each other. They wanted to make, let's say, the alliance government. What is the benefit for me? What is the benefit for us? Can anyone give another example of mutualism and symbiosis? Yes, anyone, you are all a master students. You should know about the example of mutualism and symbiosis. There are many examples. Please have a discussion, not to just to see listening and just to seeing the slides. Please. Hello. Devjit, Sagar, Rabia, those who are present.
Hello.
Yes, go ahead.
Sir, can you repeat the question, please?
What is the mutualism and can you give the example? What is symbiosis? Can you give the example?
Hello, sir.
Go ahead. Jafar, go ahead.
Yes, sir. So symbiosis, we can give an example like nitrogen fixing bacteria, which fix the nitrogen in the leguminous crop.
Can you raise your voice because I'm not able to hear you?
Okay. Am I audible, sir, now? So in symbiosis, we can give an example of like nitrogen fixing bacteria fixing nitrogen in the leguminous crops, in which the bacteria maintain close association with the root nodules, and the bacteria will derive the nutrition from the roots, and it will just fix the nitrogen for the crop, and hence they can both benefit from each other. So this is an example of symbiosis, we can say.
Okay.
Yes, sir. Yes, sir.
Okay. So what is the benefit for us?
So benefit for us will be like the nitrogen, which is the important component, the protein, which is an important component of the pulses of the leguminous crops. So that is actually obtained from the nitrogen, which is being fixed in the simpler forms by the bacteria. So it's a main component of the protein. So we get to have the proteinaceous crops by this nitrogen fixation, I guess, maybe the reason, sir.
Yes. Okay. Can anyone, others, please respond? Can you give any example where we can have the symbiotic example or mutualistic example where we are getting benefit? Or can you just understood that mutualism and symbiosis means just benefiting with each other? I think it's not. And that is where we have to understand this concept, because whole EPNE concept is based on this, the mutualism and symbiosis. Anyone wants to say something on it? What is the legume and the rhizobacteria? What kind of association they have? Yes. So basically in EPN, we have one nematode aspect, which is yesterday I have told you about the very vermiform body style. Let's say it's a multicellular organism which have distinct organ system, that's digestive system, nervous system, reproductive system, very distinctive sensory system. So that means the fully successful adapted animal, right? And the another is the bacteria. The bacteria are the prokaryotic, unicellular. They are not evolved to the any organ system, only the system where they are able to manage by the single cell only. So this is the mutualism means, and symbiosis means there is a slight difference between mutualism and symbiosis. Mutualism, and what the people have, I think Jeevan and the Sagar already told that the mutualism and told us that they are benefiting with each other. That is a mutualism. So when both are getting benefited, but when they should come first in association, right? I will give an example for your understanding, that if suppose in any one state, suppose the election happens and none of the party receive any major mandates, or they did not able to reach up to the suppose the majority mark. So what they are actually doing? They are doing alliance. So alliance they are doing, but for that, what the terms and conditions are there? That you will be on this post, you will be the chief minister, you will be for this year's, you will be for chief minister for this year's, you will be for. So these are some terms and conditions are there. So this is just a mutualism, right? So suppose anything happens in that the management, sometimes and I think that we have seen this such kind of stories, that this party has withdrawn the support from the major party, and then the government was collapsed. So the mutualism, let's say it's a temporary arrangement on the basic terms and conditions. But if you read the symbiosis very carefully, the symbiosis is not the temporary. Symbiosis means is a perfect association. It's a permanent association of two organisms, and they are benefiting also with each other, and at the same time they are never live alone with each other. So just like a promise to each other that I will not leave you in any conditions, and the same promise they receive from the other partner. So the both partners here are very important. So in EPN also is very, very important. EPN is the nematode. The nematode harbors this bacteria. Let's say harbor means if they use this bacteria in their body, and for the purpose is only that they are benefiting with each other. And what is the benefit for us? Actually, this alliance, the partnership, this associationship of these two organisms. One is a multicellular nematode, one is a unicellular bacteria. Both comes together for a common purpose, and that is to kill the insect. So EPN, please remember, it's not only the nematode, it is a package. It's a wrapped package, that the nematode plus bacteria. It's an association. And how they are actually working, I will tell you later on, but this, please remember that this is a close association. They have exchanged some of the terms and conditions in such a way that they are working for the common purpose. The purpose is here common. And you can have several examples. If you go on Wikipedia, thousands of examples are present in our nature for the perfect symbiosis. Please remember, legumes and rhizosphere bacteria are also perfect symbiosis. One are getting benefit from each other. Plant is getting nitrogen and bacteria is getting food. But how they are actually exchanging this? This is not just that just they are benefiting from each other. This is not the temporary arrangement. This is in a perfect match, we can say. It's just like normally in a marriage, we will see 36 spoons should be matched with each other, like that. They are actually, these both partners are close enough. They have associationship is such a close that they never leave each other in any condition. This is the perfect symbiosis, and in EPNs you have to see, or we will see, that this is the perfect associationship, and the purpose of this association is this, that both partners have common goal, and common goal is to insect. Nematode's goal is here to kill the insect, to eat on the dead insect. Bacteria role, the role of the bacteria is also same, to kill the insect, for benefiting for each other, because both have same goal. If they both have same goal, through the evolution, through the 1,000 years of evolutions, they come closer. So this is, please remember, EPNs is not only the nematode, it's the EPN means EPN nematode plus bacterial combination. So don't forget it is. Now I have here mentioned that there is a mutual alliance with the bacteria, which is the perfect symbiosis. And you can see here one of the dead insect is here, and it is a dissected here, and you can be able to see in this petri dish, this is a lot of nematodes are there. And here you can see in the body of the insect, these nematodes are there. So they are very safe to the ecosystem and non-toxic. Just I told you about any biocontrol agents. The first criteria for the biocontrol agent is they are safe to the ecosystem. They should be safe to the ecosystem. They should be safe to the animal. They should be safe to the human species. They should step to the any non-target organism. Then and then they can qualifies for the biocontrol agents. And this was first discovered. A very old discovery is there in 1923. So next year it will be the 100th year, we can say, for after the discovery of the EPS. So the Steiner, the name of the scientist who has discovered this, the first nematode in the, I think, Japanese beetle, and he named the name of that nematode was a Steiner nema on the contribution made by the Steiner. Same way, the another nematode, that is heterorhabditis, that is discovered by Poynter in 1977, and these now two genera nowadays we are actually using for the as EPN to control the insect pests. Now you can go for the biology and the life cycle of these nematodes. So let's say the nematodes are yes. Okay, we go for. So the life cycle is here is very simple. The nematode, yesterday I have shown you that these are the nematodes. Basically, they are in the juvenile stages, or let's say vermiform stages. They enter into the insect host. I will show the, yes, this is the life cycle. So these are the nematodes. Let's say I'm showing you here, these are the nematodes. Inside their nematode body, the bacteria are present. So once these are nematodes present, let's say in the soil, once they approach or they come in contact with the insect, they take entry inside the insect through the insect's openings. So there are insect openings are there. Let's say spiracle, mouth, anus, or let's say any body, even through the impugment also, insect impugment also, they nematodes able to penetrate inside their body. And after entering into the body of the inside the body of the insect, what happens? The nematode regurgitate. Let's say nematode vomit out this bacteria into the hemocoel of the insect body. So this bacteria actually attacks on the hemocytes of the insect, and that brings down the immune system of the insect. And that is the here, the very much quality of the EPN is that very rapid kill, or very quickly they kill the insect. So what I told, the nematode enters into the insect body and they regurgitate the bacteria. The bacteria attacks on the hemocytes and the other portions of the insect. Let's say the fatty tissues are there. Let's say some of the proteinous tissues are there. The bacteria actually secretes enzymes. Let's say the peptidase, lipases, proteinases, crystal proteins. These enzymes are released by this bacteria, and these bacteria actually brings the death of the insect. So who is here? Lethal tormentor, or who is here? The killing agent to kill the bacteria is here. The bacteria to insect is the bacteria. So this bacteria kills the insect. After killing the insect, you can see here the nematode feeds on the dead material inside the insect body. Whatever the dead material, whatever the proteinous mass, hemocoel, and the fatty tissues, whatever the content is there, whatever the dead material is, then the nematode feeds on that material. And during this process, during the feeding, because once the nematode feed, the nematode grows. And when nematode grows, molts, that molting is there, they change to the next generation, they become male, they become female, then again male, female mate with each other, they lay the eggs. This all biology happens inside the insect body. And they complete the 2 to 3 generations there. And afterwards, finally, once they got the signal that now food is depleting, now food is finishing, they change themselves into the small, let's say the juveniles. they pick up this bacteria again into their body and they release from the body of the insect through the same openings, or let's say from the by bursting the body to the opens, and they release and emerge from the body to the soil. And this is where we are actually seeing the life cycle of the nematode. Now, how specifically they have designed, you can see in nature how this mathematics they have arranged in such a way that, let's say, nematode enters into the insect body, release the bacteria, bacteria actually kills the insect, the nematode feed on the dead insect, and after the generations, after the development, growth, or let's say the metamorphosis and generations, the nematode again pick up the same bacteria in their body and they come outside into the environment. So this is how the life cycle is going on. Now here you can see the biology that in Steinernema, the nematode, this is the name of the nematode, have association with the Xenorhabdus bacteria. So the name of the bacteria is the Xenorhabdus. Again, I'm telling you, this partnership is very strict. They never allow other bacteria to enter. Steinernema Xenorhabdus. Same is in case of Heterorhabditis, the name of the nematode having association with Photorhabdus. So this is just a partnership, very strict partnership, just like two opening players. They don't want to entertain other players. Only these two, one nematode, one bacteria. So they have associations, and you can see here the nematode, they enters into the body. Here they are become male and female in first generation. What I told, once the insect killed by the nematode or by the bacteria, nematode develops on. Nematode develops and they become a male female. In the second generation, they become again male female, and after at the third generation, the juveniles comes outside. How much days it required from entry into the insect to the emerging from the insect? It is only 8 to 9 days. Let's say 8 to 10 days. So within it, this is again the specific and very beautiful quality of the EPN, that they have short life cycle. 8 to 9 days. Within 8 to 9 days, they complete the life cycle. So, and they kill the insect within 24 to 48 hours. That means a very quick kill. The killing agent, the agent is very much rapid, very much highly pathogenic, and they complete the life cycle within 8 to 9 days in case of Steinernema. Now, Heterorhabdite case, here we have one slight change in their biology. Once the nematode enters into the insect body, they become hermaphrodite. So can anybody please tell me what is hermaphrodite? If you have came across any term like hermaphrodite, hello? Are you able to listen me? Hello?
Yes, sir.
Yeah, what is hermaphrodite?
No idea, sir.
Can anyone? Yes?
Male pattern, female.
Which produces both male and female organs.
Namdev, I can't hear you. Please again repeat it. Hello, please repeat
Produces
yourself.
male and female organs presence
Presence
in
on
same
what?
individual.
Yes.
Male
yes.
and female
So
organs.
Yes. You are right. You are right. So it is you can say for rapid multiplication of the individual, the nature has given another beautiful gift like hermaphrodism. So hermaphrodism is I think majority of you have seen in entomology also, in insects, in nematodes also, hermaphrodism, this phenomena happens. And what is here, their male and female organs are present on the same individual. What is the use of it? Why the nature has changed the life cycle? Like in case of Steinernema, you have seen the male females, both are there. Can anybody have any thought why hermaphrodite is useful? Why hermaphrodism is there? Because in nature, in ecosystem, nothing is waste, right? So anything has certain modification, means certain has its utility, certain has its usefulness. So why hermaphrodism is there? Can anyone? Namdev, Jivan, if you are able to listen, Me or anyone other, please, because these names are visible here. That's why I'm taking the name. Anyone, why hermaphrodism is have given in any insect or in any organism? Why hermaphrodism is there? Why? Hello. Yes. Hello. Hello. Hello, Ashwini, Sagar, Zafar. Any idea why hermaphrodism is have its utility in any individual? Because this same question they ask in one of the exam, in ICAR exam, that's why hermaphrodites have utility. Why it is important in any organism? Why?
For rapid multiplication.
Yes, of course, for rapid multiplication. Any other guess? Of course, because then the individual who have become a hermaphrodite, they need not to go and search for their search for the partner, for a male or female. They have their own organ systems and they can able to reproduce themselves and they can produce the eggs and nutrients. But again, anyone, any other thought? Why it is? It's of course it's for the, let's say, for self-fertilize and self-propagation. Let's say we can say, but why again? So why it is important? Yes, Namdev, you are right. You are on the right track, but you are very close to the answer.
Sir, if organism produced from hermaphroditism, it contains same character, same hereditary character.
Exactly. That is the genetics here plays a very prominent role. That those organisms placed from the self-fertilization, they have their own characteristics from their single parents only. So there is no genetic recombination, I would say. I think I'm using this terminology, but how many of you understand this terminology? I don't. There is no recombination. So hermaphrodism means it's the exact Xerox copy of one parents to the other parents. And why it is important, I'm telling you. Why again it's important? Why is only the exact copy of the genetic material from my parents to me? If I am suppose the exact copy of my parent, so what will be the utility and what will be the demerit? Both you can see. I think you understood my question, because this is here the actually whole things change in the heteropdictis. Genetic makeup. Let's say I am from my parents and I am the progeny of hermaphrodite parents. Let's say. So what is the genetic makeup and what is the usefulness for me? Can anyone? Yes, Jeevan. Yes, Namdev.
Sir, can you repeat the question?
Hello. What is the utility of the genetic makeup of hermaphrodite? Because they are from the single parent, right? So what is the utility for that organism? You said they for the rapid multiplication, that is right. They are from the same genetic makeup, that is right. But what is the utility? What is the useful for us? Any thought?
It helps to increase the adaptability under undesirable conditions.
Yes, that's also true. That's also true. Again, any other thought? Ashwini, Zafar, how many are there? I don't know. I should not say the name. Priyanka, Kirtana, Rabia, Devjit. How many people? Atul, Nitin. Speak up friends, because if you are wanted to study something, you need to know the terminologies, because all your answers, I'm telling you, in ICAR examinations, those are objective answer, type of question papers are there. The answer is hidden in that 4 objectives. If you are not open up by your all senses, you are only reading and reading and reading. That does not help you at all. I'm telling you, you have to discuss instead of reading. Discuss, because from that discussion, your all senses will be open up. Until and unless you will not able to see that from that 4 objectives, 4 options, what I am having, the answer I have to select. Every time it is not happens, that only straightforward question is there and straightforward options are there. Certainly they have changed, they have turned the questions in such a way that you won't able to, you will confuse between 2 options, just like a 50-50 in KBC. And in these situations, you have to open up your all senses, that discuss it. At least after discussion, you will get something new. So hermaphroditism, whatever the people have said, it's right, that they have the adaptability to the any distant, or let's say any extreme conditions. So that is why the heterorhabditis here, once the insect is dead, food is plenty, food is enormous, they wanted to change into the hermaphrodism, because if anything happens, let's say any dry condition arrives, or let's say any wet condition arrives, or anything happens, the hermaphrodite progenies should survive in that situation. And that's why the hermaphrodism is most important as a first generation only. Because in hermaphrodite, again I'm telling you, male and female organ systems are on the same individual. They self-fertilize and they lay the eggs inside their body. From that body, the juveniles come out. The second generation will be again normal as a male female, and after the juveniles will emerge. So here for hermaphrodism, the life cycle completes within 9 to 13 days. So please remember, the modification in this biology is only for to contain, to survive in any hostile condition. Let's say any condition comes to them, their life cycle should not be disturbed. So this is all about the hermaphrodism. Now, here you can see Stenor nema killed Galleria. This is from my lab. These are the Galleria insect. I think everybody knows the Galleria, greater wax moth. This is the paste of the honeycomb. So we are rearing, and even you can see at the backside, this lab is only we are rearing the Galleria insect, and we have released the Stenor nema nematode on them. You can see the color of the cadaver. It's become yellow or yellowish to the brownish to the black. So this is the again peculiarity of the Stenor nema Xenorhabditis combination. So what happens when Stenor nema enters into the insect body? They release the Xenorhabditis bacteria. Xenorhabditis secretes some of the antimicrobial compounds, and these compounds brings down the death of the insect, and the color of that cadaver or color of that dead body is becoming a brown, or not a brown, let's say it's a black, or let's say it's yellow. But in case of heterorhabditis and photorhabditis combination, the color of the cadaver changed into the reddish brown, or a brick red color, or let's say it's a dark brown color. So it is due to the bacterial secretions. It is due to the pigmentation, the compounds released by these bacteria inside the insect host, it causes the pigmentation. So this is the speciality, and you can able to see the clear-cut difference between when any Stenor nema, any species in the world, the Stenor nema, if they attack on the insect, the color changes into the yellowish to black, and in case of heterorhabditis, the color is changes into the brown color. There are certain nematode species I have read in one of the literature that they once they attack on the insect, the color changes to green also. But only very few species are there. So this color pigmentation is there. Can anyone have any thought why such color pigmentation will be there? Why can't only they no, they have to just kill the insect? Why they have to paint this? Why they have to secrete the compound so that the color of the body has to change? Any thought? Why it is every aspect have certain scientific evidence. So every aspect have some backings of the scientific proof, or let's say I have some thought. Why the color is there? So why there is a color difference is there? It is to show to somebody. It is to show to the human beings. It is to show to someone. Why color thing is there? Can anyone, Namdev or any other people who are there, please respond. Why coloration is there? Why pigmentation and color is there?
Sir, presence of bacteria.
Yeah, of course it is due to the presence of bacteria, but why?
Pigment.
Yeah,
I
of
think.
course. Yeah, it's a pigmentation only. It's a compound released by the bacteria, and that compound, once produced in a huge quantity, they spread all over this insect body. Then it's okay, but why? My question is why? Why the coloration is there? Why? And this is a question for you. Why? Now you can say, sir, I don't know why nature has given them such quality. How should I know about it? It's not like that. Why? Okay, we will keep this question for a few minutes at the end of these presentations. Okay, now please remember the Stenarnema, once they causes the infection, or the Stenarnema infect to the insect, the body becoming black to yellow to black. And when the Phototrabidus and Tetrabditis, when attacks on the body of the insect, they become brownish red or brick red color. And I'm telling you, my friends, now you are going to you will going into the career options with you may join the private factories, you may join any private enterprises, or you may join any government institutions, or you may join anywhere. If you are working for the farmers, if some company is claiming that the insect color due to the nematode infection is due to something brown, something saying something different, please restrict that, because these qualities are very much promising. These qualities are permanent. If Tetrabditis is there, definitely the color will be the brick red. Stenarnema is there, the color is become the yellow to black. Now here you can see the hermaphrodite. Now I have clicked some of the pictures, because why I'm showing these images to you, because it will be more and more familiar to you, that this is the hermaphrodite generation of the EPN. This is Heterorhabditis hermaphrodite. Now you can able to see here, this is at the middle of this. Could you able to see this? What I'm pointing here?
Yes,
Hello?
sir. Yes, sir. Yes, sir.
Yeah. Yes. This is the female reproductive system. Let's say it is a vulva. It at the middle of the body, and at the end here you can see there is a male reproductive system. So female and male is present on the same body, and these are very large size hermaphrodites. This is the first generation is present. Now, the another one phenomena in EPN is known as endotachia matricida. This is one second. This is endotachia. This is okay. So endotachia matricida. So can anyone please tell me what is endotachia matricida? Have you came across any terminology while reading the literature? What is terminology known as endotachia matricida? Please respond. Hello? Have you came across any terminology like endotachia matricida? No? Endotachia matricida means development of the juveniles or development of progeny inside the body of the female, and the release of this progeny from their body after the death of the mother, or let's say the death of the parent. So you can see here, this is the large nematode, which is hermaphrodite nematode, and inside the body of the nematode, there are several juveniles, or let's say several progeny. The next, let's say there's a baby nematodes are present inside their body, and these nematodes will emerge from the body of the nematode, the body of this big nematode, and they first feed on the whatever the content of their mother, whatever the content of their parent, and this is the phenomenon known as Endotracheal matricida. This phenomenon is very specific, very peculiar, present in the kingdom of Animalia. To again the question is why? Because for rapid multiplication and to survive the unfavorable conditions also. So this is the Endotracheal matricida. Now there is a denticles on the edges. You can see here this is the head portions. You can see here the head portion. Here you can see the denticles. Denticles means just like a tooth-like morphological adaptation to enters inside the insect body, because insect integument is such a hard and such a sturdy. To pierce these such kinds of hard structure, there is the insect integument. The nematodes is adapted their mouth portion in such a way that is to pierce inside the insect body. So this is the denticles are also present on them. So this is the pathogenicity I have told about it. This is the nematodes enters into the insect host, release the bacteria that causes the death of the insect. Then there is the development of the nematode by feeding on the whatever the dead material present inside the insect body. There is a complete nematode complete the generations inside the what I told that first generation male female, second generation male female in case of Stenoderma, first generation hermaphrodite, second generation male female in case of the heterophrodites, and after completion of the generations, they come outside the body of the insect, and there is emergence of juveniles is present inside. Now this is the very much important, and here I wanted to I have reached to the last portion of the slides and the presentation, that is what is the terms, what I said in a perfect mutual symbiosis, or let's say perfect symbiosis, where what kind of terms and conditions on which they come close together. So here is one nematode, one is a bacteria. So why nematode is depends on bacteria, why bacteria is depends on the nematodes. So please see the slide. I have given some of the points. So nematodes depends on bacteria for what? So why nematode wants the bacteria? First, to kill the insect, because nematode wants some of the shooting agents, I would say, or nematode wants the gang of bacteria. So this is why? Because to kill the insect, because nematode don't have any kind of the killing agent or killing apparatus, let's say. Nematodes only take up this bacteria in their gut and enters inside the insect body. So to kill the insect, they need bacteria, number one. Second, favoring the environment for nematode development. I told you that once the insect is killed by the bacteria, nematode feeds on the whatever the dead material is there, inside the dead material is there. So bacteria is required for favoring the environment for the nematode development. See how beautifully they have placed have partnership in this way. First, they kill the insect. So what suppose let's assume in a very pictorial way. Let's say the nematodes ask to the bacteria, please help me for killing the insect. Then bacteria said, okay, I will help for you for killing the insect, but in return, what you are giving? So first they kill the insect. You just kill the insect for me. You develop the environment for my development. Let's say I'm a nematode, so you develop the environment for my development. Third one, sometimes if the insect, because Galleria, white glove, termites, sometimes the insect body and sometimes the insect food is not enough for the nematode. Nematode sometimes feeds on these bacteria. So bacteria serves as a food source for them. The fourth one, and which is very much specific, that bacteria is required to prevent the secondary microinfection. Let's say when nematode and bacteria are present inside the insect body, other fungi, other bacteria, other animals, they won't able to infect, they won't able to grow on the body of the insect. So this is only the playground of the nematode and bacteria. Only both will play inside their body. No other will enter it. So who is preventing them? The bacteria is preventing. A bacteria secretes antimicrobial compounds, let's say antibiotic compounds, like they say. And because of the secretions of this, all secretions of this bacteria, what is helpful is in this, that there is a secondary microinfection, like let's say fungi, let's say bacteria. There is no kinds of infection is there. So it prevents the secondary microinfections. And the fifth point for which the bacteria is helping for the nematode to breaking down the host tissues, because insect is such a multicellular, such a multi-tissue animal is there. So whatever the tissues inside the tissues are there, these are all liquefied, all are to be converted and breaking down into the simpler compounds for the nematodes. So it is just like a bacteria is feeding to the nematode. Bacteria creating the environment for the nematode development. So in return, why bacteria depends on nematode? First is to protection from the external hostile conditions. Because these bacteria, you will not get these bacteria outside the environment except nematodes. So these bacteria wanted to stay only inside the body of the nematode. They don't wanted to stay outside in the external environment, because the external environment is such a hostile environment for them. So for that, the protection is number one. So you give me the protection, I will kill the insect for you. Second point, translocation and entry inside the insect hemocyte, which is the most important, because bacteria are immobile. They don't have the that much kind of mechanism to run behind to the insect and kill them. So here, who is the agent? You carry me in your gut. You carry me in your gut, I will kill the insect for you. See, the kind of association you can see, it is just like a two gangs are working for killing one of our susceptible insects. And the third one is inhibition of antimicrobial proteins. So inhibition of antimicrobial proteins means, see, inside the insect also, the immune system is there. And every immune system have their own fighting cells. So sometimes the fighting cells are also working against to this complex, nematode-bacterial complex. During that situation, nematodes sometimes secrete some of the enzymes that will help to escape this bacteria from the attack from the immune system. So this, you can see the association. This is what I said earlier, a perfect symbiosis. So who is working for what? So nematode depends on bacteria for killing the insect, favoring the environment, prevent the secondary microinfections, breaking down the host tissues. While the bacteria depends on nematode for what? The protection from the outside environment, carrying the bacteria to the insect hemocyte, and inhibition of the antimicrobial compounds. And because of this, between the association between them, the terms and conditions, what they have written 1,000 years ago, and that's why they came closer, very much closer, so that they never leave each other in any situation. They help to each other for if suppose one partner is in a hostile condition, other partner will help to that to save my partner. So such kinds of association is there, and because of this association, the insect is killed within very quickly, and that's why the EPN after BT, after Bacillus thuringiensis, nematodes, entomopathogenic nematodes are one of the best biocontrol agent in integrated pest management, or let's say biological control. So this is the associations between the two organisms. So how to isolate this EPN from the soil? I have just mentioned this slide. So you can just bring this soil from the field, and you have to bait this soil with the Galleria. You have to put this Galleria inside the soil and wrap the beakers with the cloths and keep these beakers in the laboratory. After 6 to 7 days, if you find such kinds of colors, because I said is only the color is a differentiation is there. So if you find these colors, you put them on the dish, and here after the 6 to 8 days after the life cycle, these nematodes will emerge from their body to the water, and we will be able to get these EPNs from the soil cells, right? So this is a methodology here, and this is here you can see these are the white grubs, which is killed by the EPNs, where we are actually working on this aspect of mass production of these EPNs with help of, because white grub is just such a dreaded pest. I'm telling you, my friends, in the sugarcane, last year, I think 2019, before this lockdown, 2019, 2018, 18 and 19, this year was a very troublesome year for the sugarcane farmer, particularly in the western Maharashtra, Sangli, Satara, Solapur, Kolhapur, and Pune, Nagar, where the white grub is such a great pest. People are using the expensive chemicals, and because of the toxicity is increasing in our field. But EPNs are working very well, and with certain dose and certain formulations is needed to you apply into the field. The EPNs able to search the white grub at whatever at depth, and they able to infect, like what I told about the biology. And after the death of the insect, you can see here, this is the white grub, your third instar of the white grub. Let's say not only the second and first, but third instar, which is a very much damaging, very much harassing feeding on the sugarcane roots. So these are also killed by the nematodes. You can see these are some of the photographs. And also you can able to see, I will unshare, this is the end of my presentation. I will stop sharing. And now you can able to see here, I will I will show you, I think how you are able to see. Can you able to see this? I don't know how the background line is there. See here, you can see these are the galerias killed by the nematodes. You can see the color, the brown color is there, and this is the strain which we have isolated from the Maharashtra soils, and we are actually mass producing this EPNs on the Galleria. So you can see here, this is by this method, we are actually mass producing the EPNs on the Galleria, and we are giving to the farmers. So I think you understood that this is the EPN story. Very specific, very beautifully. They have their natures have given the gift to them to control the insect. Their target is to kill the insect and to proliferate inside the insect, and then again emerge. So these kinds of, this satisfy all the qualities of biocontrol agents. That once they are safe to the non-target organism, they are recyclable, reusable, because once you release them into our field, they can able to propagate on another insect also. So reuse and recyclable is also there. The third one is that they are very much sure, because they are killing the insect within 24 to 48 hours. They are safe to the other non-target organisms. And most important, the EPNs are easily been used by the farmers, by the cultivators, by those who are the stakeholders are there. They're easily being used into the field. So this is all about today's EPN lecture. So any doubt, any question, any curiosity questions, because I think I don't know how much I have convinced you, but whatever the topic which is very close to me, I have tried myself to reach my thinking, my thoughts to up to you. I don't know how many of you will now have the questions in their mind. Please, any question is there? Hello.
Yes, dear participants, please ask the questions if you have any. Otherwise, you can type in the chat box, but it will be better if you could directly ask to you,
Yes,
Dr. Yogi.
How much they understood the concept, I also don't know whether it is they understood or not. It is not tough. It is not that much of like statistics and other concepts. It's very easy, and actually we are working on this aspect. And but it is the most important for any crop protection pupil to know about the EPNs and what is their role in insect pest management. Yes, any question? Hello?
Introduce